Hello everyone, welcome into this session. So here we are going to discuss the few gate questions that have appeared in life sciences exam. So today's topic is chromatography and SDS-PAGE. Let's take our first question. So this question I have taken from GATE Life Sciences 2014 question paper from the biochemistry section, and this is question number 2. So I'm going to read this question first. So the question states that a mixture of arginine, phenylalanine, and histidine was fractionated using cation exchange chromatography at neutral pH. The amino acids were eluted with an increasing salt gradient. Identify the correct order of elution. So the options are given in different order. So before talking about this, let's first discuss about the basic chromatography techniques. So uh, all the amino acids form a polypeptide. These polypeptides form primary, secondary, tertiary, quaternary structure and go through a process called protein folding. And after the protein folding, the protein gets folded. Okay, so in the folding process, in order to remain soluble, the outside residues of the proteins are the hydrophilic residues, and in the core of the protein we get all the hydrophobic residues. So this is also called as hydrophobic core. Okay, and on the side we have all the polar charge residues that provide the protein the net charge. We have, so based on the hydrophobic core, their charge, their size or surface area, and their affinity towards certain chemicals or beads, we have several methods to purify our protein of interest. So suppose I want to purify the protein based on the size or its surface area, I will opt for the option of gel filtration chromatography. Okay, for the size I'll opt for gel filtration chromatography, because gel filtration chromatography sorts the protein according to their size. If I need to purify depending upon the charge, then I'll opt for the ion exchange chromatography, because ion exchange chromatography uses the uh, charge of the protein in order to purify or separate the protein from a mixture of proteins. Similarly, affinity, there are several affinity columns are available. So if your protein contains a particular residue that has affinity towards one particular bead, for example, we do the nickel anti-A purification where the protein has 6 histidine residues which bind to the nickel anti-A columns. And similarly for the GST protein, we do the glutathione saccharose binding and so on. So here it has been talked about the ion exchange chromatography. So ion exchange chromatography, as I mentioned, it utilizes the charge of the protein. Okay, so based on the charge residues, the protein can be either positively charged or negatively charged, or non-polar or polar but uncharged. So these are the different categories of the proteins. Okay, so if your protein is either positively charged, negatively charged, according to that you have two types of ion exchange chromatography. So you have cation exchange chromatography, you have anion exchange chromatography. Okay, so in this chromatography you are doing nothing but allowing your protein to bind to the uh, bead matrix which has a certain charge. So suppose if your protein of interest is positively charged, it is positively charged, you will prepare a matrix in such a way that it has negatively charged matrix. Okay, and then you allow your protein to bind here. Okay, so as the matrix is negatively charged and your protein of interest is positively charged, it will bind to the matrix, and all the non-specific proteins, either polar, non-polar, they will be removed. So suppose you have a this mixture of proteins. Okay, and now if your aim is to purify the positively charged protein or this highly positively charged protein, so in order to do that you'll perform the cation exchange chromatography. So here what will happen, as the B matrix is negatively charged, your these mixture, in this mixture only the positively charged one will bind to the matrix. Okay, suppose this one is binding, this one is binding, so the one which is having higher charge, that will bind to the more tightly to the matrix, and the one which is neutral and negatively charged, that will not bind to the matrix. Okay, okay, so when you will elute with the certain buffer or certain salt concentration, so for elution what you will do, you will again increase some positively charged ions. Suppose you will increase sodium ion concentration in the matrix, so in that case other uh, the positively charged associated proteins will be forced to come out from the matrix because sodium will go and bind there. So that will happen later. So before onwards, the unbound proteins will come out. So this negative non-charged and negatively charged will come out first, and when you start increasing the salt concentration slowly, so first the loosely or the less positive charge will come out, and then the highly positive charge, because it will take more salts to be uh, replaced by the positive sodium ions. Okay, so this will come in the last. So that is how you'll separate these cations, you or positively charged ion, using cation exchange chromatography, and opposite of that happens in the anion exchange. So in anion exchange your protein of interest will be negatively charged, and the negatively charged, the matrix which you will be having is positively charged matrix. Okay, which is initially coded with negatively charged, and when you put the mixture of protein in this column, this, okay, so the positive charge though won't bind, and neutral charge they won't bind, they will leave as you take the first elute, but these one will exchange. That's why it's called exchange. Okay, so these, this thing will be uh, separated, the already bound anion will be separated, and your particular protein of interest will go and bind. Okay, and first, and then first dilute you take, this unbound thing will come out. Okay, we'll have a situation like this. Only these one will remain, others will come out, and now in order to separate these you will increase the salt concentration, where here your salt will be chloride ion, so they will compete or replace these uh, charged proteins, and they will come out in the elution. Okay, so that is how this cation and anion exchange chromatography works. So they are also in the uh, cation exchange. So there are strong cation, weak cation columns. Okay, so there are in cation you have sulfonic acid column, carboxylic acid columns. Okay, sulfonic acid columns, carboxylic acid columns. Okay, so these are the your cation exchange columns. Okay, cation exchange columns for positively charged amino acid, but they columns are negative because in order to bind positive they should be negative, whereas these are acids, so these are the cation exchange column. So based on that we have like sp sp columns. Okay, sp sephardex columns, sp sepharose, cp sepharose, cm sepharose, all those columns will have. Okay, and similarly for anion exchange, we have the matrix of primary, secondary or tertiary amines. Those are like some weak anion exchange and strong anion exchange columns are of the quaternary amines. Okay, so there also you'll get these the Sepharose columns and everything. So these are for ion exchange chromatography. Okay, so now we'll come to our question, which is asking about a mixture of arginine, phenylalanine, histidine was fractionated using cation exchange. Okay, cation exchange chromatography at neutral pH. So the amino acid were eluted with an increasing salt gradient. Identify the correct order of elution. Okay. Huh. So at neutral pH this has been performed. So very first thing we'll see that here we are talking about a cation exchange. So cation exchange means your matrix is negatively charged. Okay, your matrix will be negatively charged, and in your mixture you have three amino acid, arginine and histidine, which are positively charged amino acid, and phenylalanine, which is non-polar neutral, no charge amino acid. Okay, so if we'll talk about the elution order, okay, it has been asked the amino acid were eluted with increasing salt content. Identify the correct order of elution. So the one which is eluted first, that will come first. So uh, the one which will not bind or have very less affinity or least affinity, that will be eluted first. So if it is talking about cation exchange, that means the matrix is covered with negatively charged ions. Okay, it is covered with negatively charged ions, and arginine and histidine being positively charged amino acids, they will bind to the columns, but phenylalanine, which is the neutral charge, that will be eluted first. Okay, so okay, so now in between arginine and histidine, which one will be eluted second? Okay, so the one which is more positive, more tightly associated, that will be eluted last. So now we have to decide even between arginine and histidine, which one is the stronger uh, base or more positively charged, I would say. Okay, so if we'll see the structure of these two, so positively charged amino acid, histidine and arginine. Okay, in histidine structure, on the side chain I'm drawing. So histidine is composed of this imidazole ring. Okay, So, okay. So this is the histidine structure. This is arginine. I'll draw. It is having 3 nitrogen atoms in its surrounding. Okay. So these are the histidine and arginine structure. Okay. So if we'll talk about the pKa value of these two amino acids. So for arginine, the pKa value is very high. Okay. It is 12.5. And for histidine, the pKa value is around 6. Okay. So pKa is nothing but the pH at which half of the copies of anything is protonated. So at pKa value, you will get 50-50 positive and negative. So at pKa you will get 50 protonated form, 50 deprotonated form. Okay. So at but the pKa, above the pKa value, you will see the pro, uh, deprotonated form dominates, and as you will go lower the pKa, okay, above the pKa you will see the deprotonated form, lower the pKa you will see the protonated form will dominate, and above the pKa deprotonated form will dominate. So if we'll see the pKa values of these arginine and histidine, so here also you'll see the resonance between these two, and here also this is a guanidine group in the arginine, where the delocalization of this charge will happens. Okay, on all 3 nitrogen, the delocalization of charge will happens. Okay, so this will have the tendency to remain in the positive form more compared to the histidine, which is having very less pKa. So higher the pKa, we can say stronger the base, or more the positively charged the amino acid. So as arginine has higher pKa, that means it can remain in the protonated form for a more in a more stable version. It will be more stable in its protonated form, and to make it deprotonated, it needs to cross the pH of 12.5. But in histidine, what will happen in histidine at so our experiment is covered at the physiological pH. So at physiological pH is around 7. So if we'll see, the physiological pH is around 7, and I have said above the lower the pKa value, the protein will remain in the protonated form. So here the arginine at pH 7, which is lower than the pKa of arginine, it will remain in the protonated form. But if we'll see histidine at physiological pH, so here the pKa is 6. So as physiological pH, it will remain in the neutral. It will have a switch in between neutral and the protonated form. Okay, neutral and protonated form will be in intern exchange. So if we'll compare the elution in between arginine and histidine at neutral pH, the first after phenylalanine, histidine will be eluted out, and then arginine. Okay, so the order of elution will be first phenylalanine, second histidine, and third will be arginine. So if we see, we have option B, which have this order of elution: phenylalanine, histidine, and then arginine. Arginine being a highly positive amino acid, having the guanidine ring in it, and having the higher pKa value, which is making it stable in positive form at the neutral pH. Okay, So this is the answer for this question. This is the NPTEL reference.